everyone, myself Dr. Roma Ghai, Associate Professor from Kite School of Pharmacy, Kite Group of Institutions, Ghaziabad, would like to discuss on the topic Comparative Observational Studies and Targeted Clinical Investigations, and this particular topic is actually a part of the subject Pharmacovigilance BP 805 ET for final year subject. So, let us start. Now, comparative observational studies. Now, first of all, you understand the word observational. Observational means in which we simply observe. The reporter is simply observing the particular experimental studies. So, comparative observation studies. So, traditional epidemiological methods are a key component in the evaluation of adverse events. As our major focus in pharmacovigilance subject is the evaluation of adverse drug reactions or adverse events. So, we have kept this particular comparative observation studies. So, these studies are actually helping in evaluation of adverse events. Observation study designs are useful in validating the signals from spontaneous reports or case series. As I have told in the last lectures, last videos, what was spontaneous reports, what was case report, was case series, these were helping in hypothesis generation. So, observation studies will help in hypothesis testing. Now, there are different types of designs. We have cross-sectional studies, case control studies, and third one is cohort studies, which include retrospective as well as prospective cohort studies. So, let us start with a cross-sectional study. A simple term of cross-sectional study means a survey. Like in survey, we collect data, right. So, cross-sectional studies are those studies where the data is collected from a population of patients at a single point in time. That means बार बार नहीं है, एक बार में ही, okay, or interval of time, maybe 45 minutes, after 45 days, after 60 days, after 3 months, after 6 months. So, data is collected at a single point in time, regardless of exposure of the disease state, exposure or disease status. It is primarily used to gather data for surveys or for ecological analysis. The disadvantage is that temporal relationship is not addressed in cross-sectional study. Let us understand temporal. Temporal means timing. Basically क्या होता है, जब हम ड्रग एक्सपोज करते हैं, कुछ टाइम के बाद हमें आउटकम मिलता है, लेकिन इन स्टडीज़ में हम टाइमिंग नहीं देख पा रहे हैं, हम ये रिलेशनशिप चेक नहीं कर सकते हैं, राइट, बिकॉज ये एक ही टाइम पे पेशेंट्स को दिया गया क्वेश्चनेयर और इनसे डाटा फिल किया जा रहा है. Right. So, these studies can also be used to examine crude association between exposure and outcome in a particular analysis. Advantage क्या है? They are best used to examine prevalence of a disease at one time point. तो हमें पता चल सकता है कितने लोगों को एक particular disease है, ये फाइंड आउट करने में क्रॉस सेक्शन स्टडीज हेल्प करते हैं. To examine trends over time when data for serial time points can be captured, we can see what is the trend over time. क्या ड्रग कब चेंज हुआ, क्या पर्टिकुलर डिजीज कब हुई, ट्रेंड्स ओवर टाइम. फॉर एग्जाम्पल क्रॉस सेक्शनल स्टडीज फ्लोरोसिस occurs by continuous leakage of fluorine in water. Now these are snapshot studies. What do you mean by snapshots? जब डाटा एक बार में कलेक्ट हो जाता है, डाटा is collected at all at once, then that is called cross sectional. So temporal relationship between the use of drug outcome cannot be determined in these studies. So, these are studies in which presence or absence of outcome or a health related variable are determined in each member of the study population or in a representative sample. एक sample ले लिए population में, उनको एक questionnaire दिया, उसमें check कर लिया क्या ये present है या ये नहीं है. तो presence or absence एकदम से हमें इस particular study से पता चल सकता है at one particular time. Exposure and outcome are determined simultaneously. Exposure मतलब उसी डाटा से हमें पता चला गया कौन सा ड्रग यूज़ किया गया है और उसके क्या आउटकम्स है उसी पर्टिकुलर सर्वे में पता चला. सो दे आर डिटरमिन्ड साइमल्टेनियसली, साथ ही साथ. These are not followed over time. All the measurements on each person are made at one point in time. Many cross-sectional studies are done using questionnaire. Alternatively, each of the subjects may be interviewed. Right? Each of the subject can be also interviewed. Next, we come to case control study. Now, this is the next comparative observation study. Now, case control study means retro. Retro मतलब पीछे. that means outcome हो चुका है, outcome has come, right? And then we are going back to trace out. So retrospective studies where a group of patients with a particular disease or ADRs are compared with a group of patients who do not have the disease. तो compare कर रहे हैं, their histories of previous exposure or risk factors are compared. Exposure status of the two groups is then compared using odds ratio. I will tell you about odds ratio in my next slide. That is, we are trying to estimate the relative risk of disease in both the groups, exposed group and unexposed group. Patients can be identified from an existing database using data collected specifically for the purpose of study of interest. तो पहले हम क्या करते हैं, patients को identify करते हैं जिनमें disease हुआ है. We can also use disease registry, which can be used as a base for the case control study. So this will help you to find out the drug exposure of cases identified from the registry, and we can also select controls selected from either patients with another condition, maybe उस तरह के type का condition है within the registry, or we can choose patient outside the registry. तो basically case control studies, cases क्या हुए जिन लोगों को disease हो गई, controls क्या हुए जिनको disease नहीं हुई लेकिन कोई और condition हुई है. Let me give you an example. First, let's see the technique, then I'll give you the example. In a case control study, cases of disease or events are identified. Controls of patient without the disease, without any event of interest are then selected from the source population. See, controls में क्या है, disease नहीं हुई है. Controls, अब आपको ध्यान रखना चाहिए, controls को select कैसे करना है. Controls को इस तरीके से select करना है कि मतलब उनमें disease भी हो सकती है. ठीक है, जिसको हम बोल सकते हैं, they are selected in such a way that the prevalence of exposure among controls represents prevalence of exposure in the source population. उनमें हो सकती है और वो हम interpret कर सकते हैं या हम extrapolate कर सकते हैं to source population. If safety information is sought for a special population, then cases and controls need to be stratified. Stratified means divided according to the population of interest, maybe elderly, children, pregnant women, सबके groups बनाए जा सकते हैं. For rare adverse events, existing large population based databases, they are efficient means, useful means for telling us what the drug exposed and all the outcome data in a short period of time for rare adverse events. The exposure status is then compared using odds ratio. So odds ratio क्या है? Let's understand what is odds ratio. It's an estimated relative risk of disease in two groups. Now, how do you calculate it? Just try to understand. We have some patients who are exposed to the drug, right? Let's take number is A, number of patients, right, who are exposed and they got the disease. So they are cases, and then we have C, which were not exposed to that particular drug, but still they have that disease. That comes C. Then we have controls where the drug was exposed, but they did not get disease, and we have controls which were unexposed, we did not get the disease. So how do you calculate this? So odds ratio, odds that a case was exposed, that's A upon C, divided by odds that a control was exposed, that is B upon D. So we get simply A upon C divided by B upon D. So this particular D gets crossed up, AD upon BC. So we can calculate the odds ratio. Let's try to understand with the example. World Health Organization WHO has identified 1143 patients, female patients, who had developed venous thromboembolic events. Please remember, venous thrombosis में क्या होता है, veins में clot बन जाते हैं, ठीक है, और ये WHO ने कुछ data collect किया in 21 countries. WHO selected 2998 controls. That means women, they were taking the drug, they were taking some medicines, but they did not experience this venous thrombosis, उनमें clot नहीं हुआ. So it was found, दोनों को check किया गया, उनकी history, उनके risk factors check किए गए, and then it was seen that previous exposure to oral contraceptives was then compared between the two groups. It was seen that patients who developed the venous thrombolysis, they were more likely to use the oral contraceptives as compared to the controls. So patients who developed PT were more likely to have. So case control से ही हमें पता चल सकता है कि कितना exposure outcome का basically relationship है. There was a study suggested that oral contraceptives were associated with the venous thrombolysis. कब यूज़ करते हैं हम इस केस स्टडीज़ को? when you want to investigate whether some association between drug and one specific adverse event is there. It is very helpful to identify the rare adverse events. It is very helpful to identify the risk factors for the adverse events. Risk factors can include conditions such as renal dysfunction or renal disorders, hepatic liver disorders, okay, renal means kidney, that might modify the relationship between the drug exposure and the adverse events. So, we need to find out कि क्या risk factors की वजह से adverse event आया या drug की वजह से आया. Under specific condition, case control study can provide absolute incident rate of the event. Exactly, we can find out how many people suffered from a particular adverse event when the drug was being taken, because we are comparing the cases with the controls. If all cases of interest or a well-defined fraction of cases in the catchment area captured, if we know all the cases and we know the controls from the source population, same source population is known, then an incident rate can be easily calculated. Strengths of case control study, it can be used to study multiple drug exposure. हमें cases पता चल गए हैं, controls हम select कर लेंगे, उनको compare करेंगे तो हमको पता चल जाएगा कि cases में क्या क्या multiple drug exposures थे. They can be used to study uncommon disease, जिसको हम rare disease बोलते हैं. They are relatively easy, fast, cheap in terms of data collection. Simple questionnaire use करना है और record से हम case control studies कर सकते हैं. But disadvantage क्या है? Finding appropriate matched controls may be difficult. हम exactly controls को find out करना मुश्किल है. They are prone to recall bias. जिससे हम questioning कर रहे हैं, जिसको disease हो गई है और हम सब पूछ रहे हैं, तो may not be able to remember the same facts. So people with disease remember things differently from those not affected. As data is collected retrospectively means outcome हो चुका है, event हो चुका है, disease हो चुकी है. So some information they may forget, they may not be available. So next type of study, again comparative observation study, is cohort study. Now what is cohort study? Cohort is a group of individuals having some common features like age. A cohort is a group who have something in common, remain part of a group over a long period of time, extended time. So cohort studies are studies of groups of patients having some common drug exposure of interest. Let me give an example. So let's take example of a study where a person wished to learn about benefits and risks of NSAIDs. What are NSAIDs? Analgesics, non-steroidal anti-inflammatory drugs. And we want to learn the benefits, we want to learn the risks on a population of patients who are likely to be prescribed. NSAIDs किसको दिया जाएगा? NSAIDs उन लोगों को दिया जाएगा जिनमें इन्फ्लेमेशन है या जिनमें पेन है, तो उस पर्टिकुलर पॉपुलेशन को टारगेट करेंगे. The cohort group would be defined on the basis of patients' exposure to NSAIDs. There are two types of cohort studies. Please remember, one is prospective, the other is retrospective. Prospective means forward in time, and retrospective means backwards, going backwards in time. So let's see the so these two, this is a type of analytical study. Please remember, cohort study, case control studies, they are analytical studies because we are collecting information about exposure status, यानी कि कितने लोगों को expose हुआ एक particular drug, and outcome status, कितनों लोगों में outcome आया, and they compare groups to identify whether there appears to be some association or link. In these studies, one measure compare the incidence of outcome to determine whether there is association between exposure and outcome. So let's try to understand this. ये किसको compare कर रहा है? ये compare कर रहा है एक group को जिसको drug expose किया गया to a group without that exposure. Okay, so we have exposed group and we have unexposed group. So in a cohort study, population at risk for the disease or adverse event is followed over time. That is prospective for the occurrence of the event. So let's try to understand. Such study is called prospective cohort study. Here the information exposure status is also known, okay, and we are following up the patients, and then we also come to know about the outcomes. So we can calculate the incident rates easily because we know which population we are going through. So cohorts are selected on the basis of drug use and they are followed over the time. That's called cohort studies. Multiple adverse events can be investigated using same data source in a cohort study. Let's try to understand prospective. We have a cohort. Cohort means a group जिनमें outcome नहीं है, disease free है. So researchers will recruit the subjects because the observational study observe करेगा researcher. तो एक exposed cohort हो गया, एक unexposed cohort हो गया. और time के बाद हमने देखा कितने लोगों में disease develop हुई, कितने disease free रही. Disease means outcome. So we are comparing the disease incidence between the exposed and unexposed group. Unexposed में भी unexposed था, फिर भी वो disease हुई है, जो outcome हम देखना चाह रहे हैं वो हुआ है या नहीं हुआ है. तो prospective means outcome has not yet occurred. The time when we start the investigation, the outcome was not there, and we are following up them for the outcome. Example, we will try to understand with example. A cohort study compared 90,000 patients who were exposed to co-amoxiclav. You know what is co-amoxiclav? It's a combination of amoxicillin and clavulanic acid. Okay, so this compared, किसके साथ 90,000 patients को compare किया गया with 3,60,000 who were only exposed to amoxicillin. तो हमारा exposed एक co-amoxiclav था और control क्या हुआ जिनको सिर्फ amoxicillin दिया गया, standard दिया गया. Study found that co-amoxiclav users were 6 times more likely to experience acute liver toxicity. Another study was to find the risk of uterine cancer between postmenopausal women receiving the hormonal replacement therapy and not receiving hormonal replacement therapy, and it was found that HRT was associated with uterine cancer. So there are some methodological issues in this cohort studies. What are they? Selection of subjects in cohort studies, both exposed and unexposed should be selected from same source population. जब हम source population बोलते हैं इसका मतलब कि उसी particular gender plus age, same criteria के लोगों को, ठीक है, same source population. Subjects who are not at risk should never be chosen. जिनमें वो outcome नहीं हो सकता for developing outcome, that should be excluded. Sampling may be effectively sampled from hospital. That means we can take such people from hospital, we can enroll from members of community or from doctors individual practice. But problem क्या होता है? Cohort study में there is loss to follow up in prospective study. कई बार क्या है जब patients का follow up कर रहे हैं तो कुछ patients ने दवाई खाना बंद कर दिया है, छोड़ दिया है country को, दूसरे country चले गए हैं और अलग type के problems आते हैं loss to follow up. किस तरह से हम follow up नहीं कर पा रहे. Retrospective means it looks back on existing data. Here means it looks backwards, examine exposures to suspected risk. The study is done using medical records, studies done using interviews with the patients, data come from large computer databases, data comes from paper charts. आपने देखा होगा hospitals के अंदर एक paper chart बना होता है जो nurses जिस तरह से medicine दी जाती हैं, तो that is paper chart for particular patient ताकि उसको timely medicine मिले, तो वो data can be collected from paper charts, then medical records. Then health records of population can be scrutinized for exposure to the study drug and the subsequent beneficial or adverse events. So in retrospective मतलब we are going backward. That means the population जिसको हम study कर रहे हैं उसमें already disease हो चुकी है, just like in case control. So let's see prospective and retrospective comparison. Here it was studies carried out from present time into future. It is tailored to collect specific exposure data, but long wait for events to occur. They are expensive, they are prone to high dropout rates. So retrospective, that is historical, that means look at medical events from some time point in the past up to the present time. मतलब कुछ past में हो चुका है, लेकिन हम उसको study present time से शुरू कर रहे हैं, backward जा रहे हैं. Advantage, information available immediately. Difficulty in tracing subjects, मैंने आपको follow up बताया. Reliance on the memory of subjects, we have to rely किस subject पे, कि subject को में क्या क्या problems हुए थे, तो हम rely कर रहे हैं उसकी memory के ऊपर कि वो जैसे बता रहा है. So this can affect the quality of the recorded information. That is a disadvantage. So advantages of cohort study, number one, we can calculate the incident rates, कितने लोगों को drug दी गई और कितने लोगों में वो adverse event हुआ. We can find out a precise risk, it can use to study multiple outcomes. They provide unbiased drug exposure data. Selection bias is less likely than in case control study. Case control study में क्या था, controls हमें select करने पड़ रहे थे, यहाँ selection bias है ही नहीं. Disadvantage क्या है cohort studies? They are relatively more expensive. Bias means anything which is giving away from truth, which is moving us away from the correct results. Okay, right? Yeah, right. So they are relatively more expensive, especially the ad hoc studies. Ad hoc studies are nothing but prospective studies. It's difficult to gather a large number of subjects if cohort studies are done prospectively. It will take years to complete. कोई ड्रग लिया है और उसके हम लॉन्ग टर्म देखना चाहते हैं, कितना टाइम लगता है लिवर टॉक्सिसिटी होने में, कितना टाइम लगता है, कितने लोगों को किडनी प्रॉब्लम्स आई, तो वो तो बहुत टाइम लगेगा ना वो डाटा कलेक्ट करने में. आउटकम डाटा में बी बायस्ड स्पेशली इन द रेट्रोस्पेक्टिव स्टडीज. जैसे मैंने बताया रेट्रोस्पेक्टिव स्टडीज़ में outcome can be maybe such type of data we are getting from the patients which we cannot rely also. Now coming to the calculation of relative risk in cohort studies, simple relative risk ratio is calculated. Relative risk ratio, what is this? It's a ratio of probability of an event occurring, that is developing a disease or being injured in an exposed group to the probability of event occurring in the comparison non-exposed group. Simply we say that the incidence or in exposed group divided by incidence in the unexposed group. So it's dividing the incidence in the exposed group by the incidence in the unexposed group. So relative risk are measures of association, that is how strongly a risk factor, how strongly exposure is associated with the outcome or adverse event. तो आपको ये फॉर्मूला याद रखना है रिलेटिव रिस्क का, इंसिडेंट एक्सपोज ग्रुप अपॉन इंसिडेंस इन अनएक्सपोज ग्रुप, कितनी प्रोबेबिलिटी है चांसेस इवेंट के होने की. एट्रिब्यूटेबल रिस्क क्या है? इट इज एट्रिब्यूटेबल रिस्क मींस दिस डिफरेंस, एट्रिब्यूटेबल रेट, difference between the incidence of disease in the exposed population, that is IE, minus the unexposed. So ये risk क्या है? Extent to which the incidence of disease can be reduced if the individual was not exposed to the risk factor. मतलब कितना extent है जो incidence को हम बचा सकते हैं, reduce कर सकते हैं, that is attributable risk. So it measures how much the trait could be prevented if that risk factor is removed from the population. So the difference in the disease rates in exposed and unexposed individuals. It's calculated from the prospective data, calculate करते हैं data से, right? Now, attributable risk is related to relative risk, but instead of it being the ratio, it is the difference. Simple. आपका relative risk में क्या था? ratio of incidence in exposed versus unexposed. और इसमें क्या है? Difference. That is the difference, right? So I have written here the proper formula: incidence in exposed group minus ये minus sign है, minus, okay, incidence in the unexposed group. Now similar to case control studies, patient cohort studies can be recognized from large automated databases. आप बहुत सारे databases हैं जिसे आप patients को select कर सकते हो, और from data collected precisely for the study. So cohort studies can be used to scrutinize the safety issues in a special population, maybe different type of population. We can do the sampling of the patients, we can stratify the cohort based on age, gender, particular characteristic, right? And there are numerous databases optimizable for pharmacoepidemiological studies. So this is a type of pharmacoepidemiological studies. Now we come to the next one, targeted clinical investigation. After seeing the comparative observation studies, when risks are identified from clinical trials, pre-approved clinical trials, further clinical studies might be called on to evaluate the mechanism action of the adverse reaction. हमें पता चल गया कि ये ADRs हो रहे हैं, तो हमें क्या करना पड़ेगा? Targeted clinical investigations. ये क्या है? Target. That means we have to find out something, some specific criteria, some specific outcome we want to find out. Here we have to evaluate the mechanism of action of adverse reaction. क्या करेंगे आप? Target clinical investigation करेंगे, trials करेंगे. In some instances, pharmacodynamics, pharmacokinetics might be conducted to determine whether a particular dose or dosing instruction can put the patient at increased risk of ADRs. कई बार ऐसे भी होता है, डोज ज़्यादा है तो हमें डोज से चेक करना है कि वो ज़्यादा डोज है तभी ADRs हो रहे हैं, या कोई डोजिंग इंस्ट्रक्शन ऐसी है, तो we have to find out we have to determine what is the risk of adverse events. Genetic testing can provide clues about which group of patients might be at increased risk of adverse reaction. Genetic mapping होती है, Specific studies can be done to investigate potential drug-drug interactions, food-drug interactions can be conducted based upon the pharmacological properties of the drug, based upon the expected use of the drug in general practice. These studies can comprise population pharmacokinetic studies. Then we have drug concentration monitoring in patients as well as normal volunteers. So, these targeted clinical investigation, they have the influence on quality, usefulness of results of the trial, right. They can influence, they can tell us what was the problem in the drug, कि वो dose की problem थी, क्या वो ADME की problem थी. These studies are large resource intensive trials. So, these are the references from which I have compiled the data. So, thank you everyone for patient listening. I hope you have understood what are comparative observation studies. We have gone through cross-sectional, case control, cohort studies, and then we have seen the targeted clinical investigations. Thank you all.